Greetings and welcome to Oral Pathology 360, the fundamental series. It's lovely to see you all here as always. And there's so much happening, but more than everything, I just wanted to remind everyone because I had some queries. The best way to take part in these sessions is if you log in using your YouTube or your Google, whatever login that is required, because then you can comment and you can also put in your questions in the chat box, which we answer at the end. And also, it would be lovely to know where in the world you're from. A lot of the work that we do at Oral Pathology 360 is about connecting people. So let's connect here right away. Let us know which part of the world you're connecting in and definitely leave a comment. Welcome to everyone who's already here now. To begin with the program. As usual, at the beginning, I got to tell you we got important news. You can stay on till the end to see that. And about today's lecture, we have on molecular diagnostic methods and by Dr. Divya Upala. Now, this is I can already see that some of our seniors have logged in, and I welcome you. And I do hope you will add to this. It is the fundamental series. We hope to cover a vast, compounded, like sort of a review of the whole thing for our postgraduate students. And to tell you a little about Dr. Divya Upala now, she is professor and head Department of Oral Pathology and Microbiology at Gandhinagar in India. Okay, that's Gandhinagar, Vishakhapatnam. Okay, right. I would have thought Gandhinagar. I just thought of Gujarat. Right. and Many of you realize she's there on LinkedIn and on various platforms, so you might be already in contact with her. Welcome, Dr. Divya. Let me bring you here so you can say hi too. Yes, you are on. I think your mic is off.
Can you hear me, ma'am?
Yes, I can.
Good morning, everyone. Glad to be here. Thank you, ma'am, for inviting me over. Thank you so much.
You are most welcome. So now let's move on. We will all watch the lecture together and then we will come back for the next part.
A very good morning to one and all. I'm Dr. Divya from Geetham Dental College and Hospital. Today, I take this opportunity to discuss molecular diagnostic methods with you. Today, I'm going to discuss about polymerase chain reaction and a few variants, various hybridization methods used commonly, DNA sequencing, laser capture microdissection, flow cytometry, proteomics, transcriptomics, and nanotechnology. Let's understand a few terminologies to know it better. Chromosome. These thread-like structures are present in the nucleus with long DNA sequences. Gene. This part of the DNA molecule directs the synthesis of specific polypeptide chains. DNA, it is the genetic material that codes for genes. Codon, three adjacent nucleotides coding for one amino acid. Base pairing, specific bonding between purines and pyrimidines. cDNA, single stranded complementary DNA. mRNA. This form of RNA mediates transfer of genetic information from the nucleus to ribosomes, serving as a template for protein synthesis. Transcription. In this process, genetic information is copied from DNA to RNA. Translation. The information on mRNA is used to make proteins. Now, coming to polymerase chain reaction. This is an in vitro method for amplification of specific short fragments of DNA. This method was conceptualized by Carey Mullis in 1985. This method revolutionized diagnostic and therapeutic ability. Its applications include and ranged from microbiology detection of bacteria, virus, parasites, and fungi. Human genetics, identification of chromosomal disorders, and other hereditary diseases. In forensic pathology, identification of mutilated or decomposed human tissues for sex determination and paternity cases. Tumor biology to detect mutations, cancer associated and cancer associated oncogenes. Requirements for PCR are template DNA, primer, deoxynucleoside triphosphate, and DNA polymerase. The method includes extracting DNA followed by denaturation of the template for 30 seconds at 93 degrees Celsius, annealing of the primer for 30 seconds at 55 to 60 degrees Celsius, extension of DNA for 1 minute at 72 degrees Celsius. The machine used in conducting PCR is called as a thermocycler. Coming to the types, they include nested, multiplex, reverse transcriptase, and quantitative PCR. Blood, urine, saliva, and feces are the samples through which DNA can be extracted and used. Poor quality or cross contamination of DNA can result in false positive or false negative. This pictograph shows the diagrammatic representation of the process. In a typical PCR reaction, the cycle is repeated 25 to 40 times and takes approximately 2 to 4 hours, depending on the length of DNA needing to be copied. The reason for this is that after the first cycle, it is no longer only the original DNA template that is being used for amplification, but also the new strands that we are used in the reaction. Additionally, we do not add just one Taq polymerase or primer to the sample. We add several to float around the reaction. So the number of DNA molecules can roughly double with each round of cycling. All forms of PCR consist of some common steps such as denaturation, annealing, and extension. Real-time PCR quantifies the starting amounts of DNA and requires less time because there is no gel-based analysis. Nested PCR uses 2 pairs of primers which can specify a particular target. This process detects point mutations with a great specificity. Multiplex PCR amplifies DNA using multiple primers, thereby rapidly detecting deletions and duplications in a gene. RT-PCR or reverse transcriptase PCR uses the enzyme reverse transcriptase, making this a sensitive application to detect a low copy of RNA. When adequate migration has occurred, DNA fragments are visualized by staining with ethidium bromide. This fluorescent dye intercalates between bases of DNA and RNA. It is often incorporated into the gel so that staining occurs during electrophoresis. But the gel can also be stained after electrophoresis by soaking in a dilute solution of ethidium bromide. To visualize DNA or RNA, the gel is placed on an ultraviolet transilluminator. Be aware that DNA will diffuse within the gel over time. An examination or photography should take place shortly after cessation of electrophoresis. Fragments of linear DNA migrate through agarose gels with a mobility that is inversely proportional to the log of their molecular weight. As the voltage applied to a gel is increased, larger fragments migrate proportionally faster than smaller fragments. For that reason, the best resolution of fragments larger than about 2 kb is attained by applying no more than 5 volts per centimeter to the gel. Several different buffers have been recommended for electrophoresis of DNA. The most commonly used for duplex DNA are the tris acetate EDTA and the tris borate EDTA. DNA fragments will migrate at somewhat different rates in these two buffers due to differences in ionic strength. Buffers not only establish a pH but provide ions to support conductivity. If you mistakenly use water instead of buffer, there will be essentially no migration of DNA in the gel. Coming up to hybridization. This is the pairing of complementary RNA or DNA strands to produce a double-stranded nucleic acid. The transfer of macromolecules such as nucleic acids and proteins to solid phase membranous support is known as blotting. In this particular section, I would discuss the various hybridization methods, applications, and limitations. A sudden blot is a method routinely used in molecular biology for detection of a specific DNA sequence in DNA samples. Sudden blotting combines transfer of electrophoresis separated DNA fragments to a filter membrane and subsequent fragment detection by probe hybridization. In oral pathology, in a study where PCR and a sudden blot technique was used, it was found that sudden blot hybridization had a positivity of around 16%. The other blotting techniques emerged from this method have been termed as Northern for RNA, Western for protein, Eastern for post-translational protein modifications, Southwestern for DNA protein interaction blotting. In northern blotting, nylon membrane with a positive charge is most effective for use since the negatively charged nucleic acids have a high affinity for them. The transfer buffer used for the blotting usually contains formamide because it lowers the annealing temperature of the probe RNA interaction, preventing RNA degradation by high temperatures. Western blot is often used in research to separate and identify proteins. In this technique, a mixture of proteins is separated based on molecular weight and thus by type through gel electrophoresis. These results are then transferred to a membrane producing a band for each protein. In this technique, DNA strands are cut by restriction endonucleases to smaller fragments which are electrophoresed on agarose gel with a nitrocellulose. The subsequent pattern of hybridization is visualized on an X-ray film by autoradiography. This pictograph illustrates the mechanism of sudden blotting. Northern blotting. A general blotting procedure starts with the extraction of total RNA from a homogenized tissue sample. RNA samples are then separated by gel electrophoresis. Since the gel are fragile and the probes are unable to enter the matrix, the RNA samples, now separated by size, are transferred to a nylon membrane through a capillary or vacuum blotting system. A nylon membrane with a positive charge is the most effective for use in northern blotting since the negatively charged nucleic acids have a high affinity for them. The transfer buffer used for the blotting usually contains formamide because it lowers the annealing temperature of the probe RNA interaction, preventing RNA degradation by high temperatures. Once the RNA has been transferred to a membrane, it is visualized on an X-ray film by autoradiography. Western blotting. The proteins of the sample are separated using gel electrophoresis. Separation of proteins may be by isoelectric point, molecular weight, electric charge, or a combination of these factors. The nature of the separation depends on the treatment of the sample and the nature of the gel. This is a very useful way to determine a protein. The protein moves from within the gel onto the membrane while maintaining the organization they had within the gel. As a result of this blotting process, the proteins are exposed on a thin surface layer for detection. Typically, the solution is composed of a buffered saline solution with a small percentage of detergent and sometimes with a powdered milk or BSA. The antibody solution and the membrane can be sealed and incubated together for anywhere from 30 minutes to overnight. It can also be incubated at different temperatures, with warmer temperatures being associated with more binding, both specific and non-specific. Comparative genomic hybridization. In this method, tumor DNA labeled with biotin is detected with fluorescent marked in green, normal DNA labeled with deoxygenin detected with rhodomin is marked in red. Both samples are hybridized together on a metaphase spread chromosomes. The region of deletion in the tumor cell hybridizes with normal DNA marked in red. In situ hybridization. In this type of method, a labeled complementary DNA or RNA is used to localize a nucleic acid sequence in a tissue. This technique is very commonly used to detect viruses in the cell and mutations in malignancies. In situ hybridization is a type of hybridization that uses a labeled complementary DNA or RNA strand, that is, to localize a specific DNA or RNA sequence in a portion or section of tissue, or if the tissue is small enough, in the entire tissue. This is distinct from immunohistochemistry, which usually localizes proteins in tissue sections. DNA in situ hybridization can be used to determine the structure of chromosomes. Microarray. In this method, complementary acid hybridization is the principle. It utilizes a probe and a target, which is a fluorescent labeled complementary DNA synthesized from an RNA sample. Microarray is most frequently used to measure changes in expression levels, detecting single nucleotide polymorphisms, and molecular analysis of malignant tumors. DNA sequencing. DNA sequencing is the process of determining the nucleic acid sequence, the order of nucleotides in DNA. It includes any method or technology that is used to determine the order of the 4 bases: adenine, guanine, cytosine, and thymine. Knowledge of DNA sequences has become indispensable for basic biological research. DNA genographic projects and in numerous applied fields such as medical diagnosis, biotechnology, forensic biology, virology, and biological systematics. Comparing healthy and mutated DNA sequences, and it can diagnose different diseases including various cancers. This pictograph illustrates the sequencing method where the ATPs terminated the polymerization chain reaction on its incorporation. Genotyping versus sequencing. Genotyping refers to the identification of variations of different strains or subtypes of the pathogen at nucleic acid level. Genotyping of PCR amplicons can be performed by direct sequencing, by reverse hybridization to genotype specific probes, by restriction fragment length polymorphism, or by detection of single nucleotide polymorphisms with mass spectrometry. Direct sequencing is usually considered as the gold standard as it provides accurate and extensive data. An example of genotyping currently moving into routine practice is human papillomavirus typing. Genotyping for the oncogenic HPV types enables risk assessment, personalized clinical management, and early intervention in women with mildly abnormal PAP results. Genotyping is also widely used for molecular epidemiology and in identifying antimicrobial resistance and high risk strains. This figure illustrates the analogy of a book being used to compare sequencing to genotyping. Sequencing is like reading out all the letters in a book. Genotyping is looking at a specific section of a specific page and comparing to others of the same page. Flow cytometry. Flow cytometry uses the ability of cells to undergo light scattering, excitation, and then emission of absorbed fluorochrome molecule when exposed to light source. Its application can be seen in the determination of DNA ploidy rates. Laser capture microdissection. The laser capture technique was developed in the 1990s by Dr. Buck and colleagues at the National Institute of Health. Laser capture microdissection is an automated sample preparation technique that enables isolation of specific cells from a mixed population under microscopic visualization. Laser capture microdissection is a powerful technique that enables oncology researchers to compare molecular profiles of tumor tissue to surrounding non-tumor cells. Proteomics. Proteomics is the large-scale study of proteins. Proteins are the vital parts of living organisms with many functions such as the formation of structural fibers of muscle tissue, enzymatic digestion of food or synthesis, and replication of DNA. In addition, other kinds of proteins include antibodies that protect an organism from infection and hormones that send important signals throughout the body. The proteome is the entire set of protein produced or modified by an organism or system. Proteomics enables the identification of ever-increasing numbers of proteins. This varies with time and distinct requirements or stresses that a cell or organism undergoes. In proteomics, the process includes separation of the protein identification and characterization, and finally, utilization of the database. Proteomics is a revolutionary technique that has been used in medicine, including drug and biomarker discovery. Proteomics can identify and monitor biomarkers by analyzing the proteins in the body fluids such as urine, serum, exhaled breath, and spinal fluid. Proteomics can also facilitate drug development by providing a comprehensive map of protein interactions associated with disease pathways. Transcriptomics. Transcriptomics is defined as a study of transcriptome, the complete set of RNA. The really helpful aspect of transcriptomics is that they are smaller than genomes. Genomes contain junk DNA, introns in genes, and structural repetitive regions such as centromeres and telomeres, which are not transcribed. Thus, they are not sequenced in transcriptomics projects. As mentioned above, transcriptomes are tissue specific, so a researcher also reduces the amount of data because they are only sequencing one tissue and the genes expressed in that tissue. Researchers may want to use a transcriptome if the genome of the organism they study is very large. Nanotechnology. Nanotechnology, also shortened to nanotech, is the use of matter on atomic, molecular, and supramolecular scale for industrial purposes. The earliest widespread description of nanotechnology referred to the particular technological goal of precisely manipulating atoms and molecules for fabrication of macro scale products, also now referred to as molecular nanotechnology. The application of nanotechnology in medicine and healthcare is referred to as nanomedicine, and it has been used to combat some of the most common diseases, including cardiovascular diseases and cancer. There has been many recent advances of nanotechnology in the aspects of imaging and drug delivery. Future directions. A new generation of programmable diagnostic devices has begun, which is taking the advantage of information generated from the study of genomics, proteomics, metabolomics, and glycomics. This biochip uses nanotechnology and has been used in the detection of oral cancer. Finally, coming to practice points. Appropriate site, quality, and timing of specimen collection are vital for reliable molecular diagnostic analysis. To fully exploit the potential of new molecular tests, clinicians need to recognize their enhanced opportunities for diagnosis, but also their constraints. Laboratory results should always be interpreted in the context of the clinical presentation of the patient. These are my references. To conclude, finally, I would like to thank my colleagues, dear postgraduates in my department I'm working at for the support. A heartfelt thank you to my teachers at Ragas Dental College for their guidance. And last but not at all the least, Dr. Mandana ma'am, to give me an opportunity in this wonderful platform for presentation. Thank you, ma'am.
Right. Thank you so much, Dr. Divya. That was an interesting presentation. And now we will look and see what questions we have. Sorry, listening from 2 years is always very confusing when you're listening to two different things. Okay, so let's see where we are. I request everyone to please put in your questions as usual. You know the drill, and we will get to them. In the meanwhile, let's see what we already have. And we'll share some of the comments till we get to the actual questions. So we have from Dr. Leela Kadam. She was wishing you, and of course, super excited for the lecture of my beloved teacher, right? And let's not forget, first thing in earlier than everybody, as usual, we had Dr. Nasser who joined us from the US. Thank you for being here as always. Then we had Dr. Niharika and Dr. Shruti. Happy Tuesday. Yes, I guess Tuesdays are memorable because of the oral pathology part in it, anyways. So, yes, it is exciting to be here too. Dr. Garima, good morning. And Dr. Afrin, good morning. And Dr. Nasser says, marvelous, practical, and superb review. Thank you for being here. And Dr. Leela, thank you for such a wonderful lecture to brush up in short. All molecular methods, very informative. Yes, thank you for your comments. So, I mean, now you heard everyone's comments. You want to say anything?
Uh, I'm sorry I couldn't complete or cover a lot. There are so much. There's so much in it, and but I could just do this much for now. Uh, so please forgive me for my mistakes. And I'm very glad if there are any comments, further comments or questions.
Yes, it will be nice if we have some questions. Anyone has a question on this? We have one more comment from Dr. Kumar. Good morning, ma'am. Thank you very much. Such an informative lecture. Had a great lecture. Yes. Thank you for being with us. And yes, Dr. Afreen also says thank you. Dr. Stacy, very interesting and informative lecture. Thank you so much. You are most welcome. Although, really, I do realize that you're saying thank you to Dr. Divya. I'm just saying thank you on her behalf. That's something. Sorry. Okay. We have something here. Yes, we have a question, I think, from Dr. Nasser. Deparaffinized sections, your preferred method for tumor mutations. For undeparaffinized sections, I guess.
The paraphenase sections, your preferred method for tumor mutations.
We lost your voice again, doctor. Right.
Actually, there's not a single one, but we can use many molecular techniques as we have seen. So, deparaffinized, we can go for this microarray, and in situ hybridization is again on that paraffinized. So there are many techniques. So I can give you a detailed answer if and I'll post it online because
Right.
there's so many of it.
Okay. So she's going to give us whatever
Yeah.
replies are going to take more time than required here, than possible here, in the comments of the YouTube. So you can all check it out. Right. Dr. Niharika also saying informative session. Thank you. So you're most welcome, Dr. Nasser. And right. So anybody else with a question? Right. Is now I also have a slight lag between what you post there and what comes on my desk here. Okay, right, it's come. So is the voice okay? Right. So there is one from Dr. Neetu. Good morning to all. Wonderful presentation, ma'am. Which molecular method will be economical and feasible for saliva studies?
There are few techniques which are there in a resource limited setting in our country that in our department itself we have used strip method um, and simple filter papers to detect the pH of the saliva. So we can coordinate with the biochemistry department and definitely we can use these type of methods instead of expensive thing. All these are very expensive in our country at present to use on a large scale.
Right. So okay, I'm not seeing any more questions anywhere. Right. I think that's all the questions we have. So let's give you your certificate. Yes, thank you very much for the presentation.
Thank you so much, ma'am. Thank you. Thank you so much.
You're most welcome. And we just got a question. Okay, let me bring it. You can answer it now only. Yeah, Dr. Osmani, uh, from from Ecuador, I think. Which is the method diagnostic for diagnostic behavior of? I think there's a little repetition there. Must be the diagnostic method for behavior of aggressive lesions. Will any of these work? You put on your mic, please.
So we have these two methods, cytogenetics and molecular diagnostics. Uh, because he has asked about molecular diagnostic methods, I'll try to answer in that. We can use the comparative genomic hybridization technique and see the differences between a normal and aggressive lesion. So we can point out the mutations in those lesions.
Right. Okay. Okay, I think we are done with the comments and everything. And now let's just go with the news that I was supposed to share with you. So let's do that. So here is the OP360 news for this week. Now, I told you last week about the histopathology masterclass, which is jointly organized by Oral Pathology 360 and Vishnu Dental College. This week, we are announcing a free giveaway entry for the event. Two entries to be exact. Now, entering the giveaway itself is easy. You got to go to the event page and click giveaway. Complete as many social media actions as possible, like follow us on Facebook, Instagram, and LinkedIn, or subscribe to our YouTube channel. Each one of these actions will give you one entry to the contest, and sharing it gives you three bonus entries. Now, to share, you have to hit the social media share icons that are available after submitting your form. Of course, many of you are already a part of our community, you're already following us or are subscribed to the channel. Now, don't worry, that doesn't mean you lose here. All you got to do is hit on the appropriate icon. It will still register that you have completed that action and you will get the points. It's an excellent opportunity to get a free entry and also to connect with us on different platforms, which was really the purpose of the whole thing. Now, we will close the giveaway on the 28th and issue the lucky entries on the 29th. Another interesting bit of news about the masterclass is that we have received 3 CD credits for the event from the Dental Council. So really there is no reason to wait. Hurry up because seats are filling up fast and they are limited as we mentioned earlier. And next bit of news is really more of a reminder that the application to the 6 month free training in social media leadership will be closing in 8 days. If you're interested, you need to hurry. And finally, a reminder that there is now a dedicated section on the website from where you can contribute to Oral Pathology 360 financially or through your suggestions for content and speakers. And also a reminder that your interaction with the content here and on all social media platform is very helpful and important in promoting the diagnostic sciences of dentistry, which is what we are doing together. So remember to like, to share, to comment. It all makes a difference. Now, to reach any of these events or even the contribution page, there are different ways you can go through it. You can see the video description below. You can scan the relevant QR code or visit the website and look at the tabs. You will be actually quite easily be able to find everything there. It should not be difficult. Still, if you have any difficulty, well, we are here to help. You can email us at info@oralpathology360.com or contact us on LinkedIn, Facebook, and Instagram by using our handle in all of these, which is common, and it is at the rate oralpathology360. Moving on, what we have lined up for next Tuesday. Right. So thank you for watching that with me. And next week we are having another lecture, a recorded lecture, which was a part of the international conference we had last year, and that is odontogenic carcinomas from Dr. Yameli Rui Vasquez. Okay, I don't get that very well, and it's a collection of rare entities and how to make the diagnosis. I do hope you will be with us for that in the coming week. And with that, well, we have reached the point where I say thank you for being with us, to all our speakers, to Dr. Divya, to everybody who takes the time and the effort to put in a comment and to take part in the chat. Really, it's what makes it all more interesting, and we really appreciate that. At the same time, I can see there are already a lot of likes, but if you did like this and you haven't still liked it, please hit the like so Dr. Divya knows you liked her lecture. And with that, I guess it's time to say bye and to catch up with you next week. See you all.